Greetings everyone. So now we are going to discuss about the DNA replication. The process occurs in three major steps. Number one step in DNA replication is origin of replication. Number two step in DNA replication is elongation. And number third step is termination phase. Now when it comes to the replication of DNA, as previously mentioned, it is DNA replication is a autocatalytic process, which means the DNA itself causes to catalyze the DNA itself catalyzes the replication of itself, due to which the DNA is able to replicate itself by its own, due to which we call this process as autocatalytic process. But if we talk about the conversion of, not the conversion, but the synthesis of mRNA by using DNA strand, the formation of mRNA using DNA, so the formation of mRNA using DNA, which is also known as transcription, that process is an heterocatalytic process. Heterocatalytic process. Now, why is it called as heterocatalytic process? Because that process involves two different molecules or two different catalysts, which the nature of the catalyst and the nature of the product is different. So that is why this is not sorry. It is not a heterocatalytic process because the nature of the reactant, which is the DNA, is different than the nature of the product, which is RNA, due to which it is a heterocatalytic process, which is known as transcription. Now, when it comes to the process of DNA replication, we have three major steps. Number one is origin of replication. In origin of replication, what actually happens is that there is unzipping of DNA. Just like if we are having a zip of let's say a bag, we are supposed to unzip it in order to open the bag. So in order to open the DNA, what we are going to do, we are going to unzip the DNA with the help of an enzyme which is known as the helicase enzyme. Helicase enzyme is the enzyme which causes to break the hydrogen bond between the adenine and thymine and cytosine and guanine. Adenine and thymine have double bond. The guanine and cytosine have triple bond, which are broken by the helicase enzyme. Now, helicase enzyme does not directly start to uh, break down the hydrogen bond from any part of DNA. That's not the thing. Helicase enzyme starts the replication by a site on the DNA, a site on the DNA which is known as the origin of replication. Origin of replication, or ORI. Origin of replication is found one in number in prokaryotes, is found one in number in prokaryotes, and it is found hundreds and thousands in number in eukaryotes, because in eukaryotes we've got a very long DNA. It's around 2.2 meter in a single what do we call it, cell. But if we talk about uh, prokaryotes, the DNA is much smaller in size, much smaller in length. So at that moment, we only require one ORI in prokaryotes and hundreds and thousands in eukaryotes. All right. And one more thing that I'd like to highlight over here is that the transcription, the process of DNA replication is process of DNA replication only occurs from side of 5 prime to 3 prime. DNA replication only occurs from 5 prime to 3 prime. It does not occur from 3 prime to 5 prime. And it occurs from two different sides at the same time, such, such that it is bidirectional. It is a bidirectional replication. It is a bidirectional replication of DNA. Bidirectional replication, what does it mean? It means that all that the replication of DNA will occur from 5 prime to 3 prime, but that would be a bidirectional replication, such that if we are having, let's suppose, a DNA molecule. Now what actually happens is that the helicase enzyme, let's say we are giving green color to the helicase enzyme. Helicase enzyme, what will helicase enzyme do? It will cause to act on, it will cause to act on DNA, um, it will cause to act on the DNA molecule and cause to break hydrogen bond from one hand side. One helicase enzyme would be breaking hydrogen bonds from one hand side, and another helicase enzyme, let's say we are marking it with yellow color, that would be breaking the hydrogen bond from the other hand side, which means at the same moment the DNA replication would be occurring from two different directions, two opposite directions. But those two opposite directions would be from 5 prime to 3 prime, due to which we call this a replication a bidirectional replication, which only occurs from 5 prime to 3 prime. Now, this is about a concept. Now, if we talk about, sorry, if we talk about the DNA replication, when the helicase enzyme acts on the DNA strand in order to break the hydrogen bond, it causes to create a bubble of replication, a bubble in which the entire DNA strand is covered. That bubble is known as the replication bubble. And when the helicase enzyme causes to break the uh, hydrogen bond between the two strands, then it causes to form a fork type appearance. A Y-shaped fork type appearance is formed, which is known as the replication fork. Just like this is a Y-shaped appearance, this Y-shaped structure is known as the this Y-shaped structure is named as the replication fork because it looks like a fork in actual. So it is a replication fork. Now, as helicase enzyme acts on the as a helicase enzyme acts on the DNA strands from 5 prime to 3 prime side, then at the same moment, the replication, at the same moment, the breaking of hydrogen bonds from one end side causes to create a tension in the DNA strand on the opposite side from 3 prime to 5 prime side, due to which it causes to create tension from 3 prime to 5 prime side, causes to create tension from 3 prime to 5 prime side. That tension is created due to supercoiling, because from one end side the DNA is being broken and from other end side the DNA is being tightly curled up, due to which supercoiling occurs and thus the tension is produced within DNA. In order to alleviate that tension, there is an other enzyme utilized over here which is known as topoisomerase. Topoisomerase is utilized for alleviating tension by removing supercoiling in eukaryotes, and in prokaryotes we are having gryase. In prokaryotes there is gryase, and in eukaryotes there is topoisomerase, right? These are the two things. Now once this is done, what actually happens is that once the hydrogen bonds are being broken in the DNA molecule, with the help of helicase enzyme, on the back side there are chances that these two strands which are apart from each other might form hydrogen bonds once again with the might form hydrogen bonds once again with their complementary strand. So in order to prevent the formation of hydrogen bonds, there is a protein utilized which is named as the single strand binding protein or single strand stabilizer protein, SSSP or SSBP, which basically causes to act as a which causes to act as a destabilizer, which causes to act as a destabilizer which causes to act as a destabilizer, a bond destabilizer, when it causes to act as a strand destabilizer or bond destabilizer, right? And this is the first step in which is known as the origin of replication. Now, when it comes to the second step, which is known as the DNA, which is known as the elongation phase. What actually happens in elongation phase? We have studied that in super conservative model of DNA, what actually happens is that a DNA strand, the parent strand, has the addition of nucleotides by the help of special enzymes known as DNA polymerase, which causes to form, you know, a complementary strand of which is known as the daughter strand of the parent strand. The complementary strand of the parent strand is known as daughter strand, which is formed with the help of DNA polymerase. Now, over here we are having three major type of DNA polymerase: DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. When it comes to the major enzyme which will act in addition of nucleotides and forming the complementary daughter strand, we would be utilizing in that major enzyme as the DNA polymerase 3. DNA polymerase 3, right? What actually DNA polymerase 3 does, it causes to add nucleotides to form a complementary strand, causes to form a complementary strand, complementary daughter strand for the parent strand which is already existing. Now, what DNA polymerase 3 does is that DNA polymerase 3 is its function is to form phosphodiester bond and the bond of nucleotides, addition of nucleotides in order to create the deoxyribe, in order to create the complementary daughter strand. So there is a problem the DNA polymerase 3 does not act directly. It cannot initiate the replication process directly. It needs to be stimulated by another enzyme, by another particle, by another fragment, which can help it get the direction for the addition of nucleotides. So for that, this property is first and foremost named as de novo. DNA polymerase 3 is de novo, which means it cannot start replication by its own. And in order to help it initiate the process of replication, what actually happens is that there is the formation of an RNA fragment, which is around 6 to 10 bases long, 6 to 10 base pair long RNA fragment, 6 to 10 bases long RNA fragment, which is known as a primer, which is known as primer. And that primer is formed with the help of an other enzyme, which is known as primase, right? So what actually happens is that before the action of DNA polymerase 3, the before the action of DNA polymerase 3, the primase enzyme acts to form a short RNA fragment, the which is known as the primer, which runs from 5 prime to 3 prime. Now what actually happens is that let me just try to draw another structure over here. Let's say this is the replication fork, right? This is the replication fork, and over here this is an strand. This is a strand which is running from 5 prime. Sorry, this is a strand. Let's say this is running from 5 prime to 3 prime. 5 prime to 3 prime, right? 5 prime to 3 prime. Let me just try to draw the structure. All right. So what actually happens is that the primase enzyme comes over here. Primase, let's say primase comes over here and causes to synthesize a DNA strand. It causes to synthesize an RNA fragment, sorry, of 6 to 10 bases, 6 to 10 nitrogen bases, which is known as the primer. And what is the major function of primer? It initiates the process of replication by giving the direction to the but it initiates the process of replication by giving direction to the DNA polymerase 3 to add nucleotides, so that it can add nucleotides in a particular direction with the help of the primer, which is already present, and thus helps in the process of initiation of replication. So initiation of replication is important because it is required to get the direction in which the DNA polymerase 3 would be adding the nucleotides. So for that, it is responsible for giving the direction which is in which the DNA polymerase 3 would be adding the nucleotides. Now once the primer is formed, then at that moment what actually happens is that DNA polymerase comes over here. Let us give a new color on DNA polymerase on a DNA polymerase 3. So let's say this is DNA polymerase 3, and over here what DNA polymerase 3 will do is that what actually happens is that DNA polymerase 3 will cause to add the complementary nucleotides. Complementary nucleotides would be added by the DNA polymerase 3, and it will cause to create a DNA, it will cause to create the complementary daughter strand, which would be anti-parallel to the parent strand and would be parallel to the primer. That would be parallel to the primer and anti-parallel to the daughter strand, such that daughter strand runs from here from 3 prime to 5 prime, while the parent strand runs from 3 prime to 5 prime. So the daughter strand would be running from 5 prime to 3 prime, which is anti-parallel to the parent strand and parallel to the direction of primer, which is from 5 prime to 3 prime, due to which when DNA replication will occur, it will cause to add nucleotides in this direction, right? I'm just trying to draw a rough structure in order to give you an idea about it, right? So what actually happens is that when the DNA polymerase creates around 100 to 200 nucleotides. When the DNA polymerase adds 100 to 200 nucleotides in a single daughter strand, it causes to stop over here, and another DNA polymerase 3 enzyme comes, another DNA polymerase 3 enzyme comes and causes to create another DNA fragment, another complementary daughter strand, which would be of around 100 to 200 nucleotides in case of, which would be of 100 to 200 nucleotides, or it can be of 1000 to 2000 nucleotides as well. 100 to 200 nucleotides in eukaryotes and 1000 to 2000 nucleotides in prokaryotes. So either 100 to 100 or 1000 to 2000 nucleotides, right? Okay. So there's a question that why does a single DNA polymerase 3 enzyme cause to form the entire nucleotide structure? So this is something. Now, if it should be a question that why DNA polymerase 3, a single DNA polymerase 3 can add all of the nucleotides without having the need, without having the necessity to utilize another DNA polymerase 3. So it is important because the DNA polymerase 3 has to, that is important because DNA polymerase 3 cannot work on the entire chain as let's suppose, because the DNA polymerase 3 would be moving from, if the DNA polymerase 3 would be moving in this direction, let's suppose, then at that moment it would be following the direction of the primer, right? But after a certain distance, there might be the possibility that primer would be far away from it, due to which it won't be able to get the direction from the primer, due to which after a certain distance there is the requirement of addition of another primer, another primer with the help of primase enzyme to help the DNA polymerase add another chain of, to help the DNA polymerase get the direction to add the nucleotides to form the complementary daughter strand. So as a result of which, after a few nucleotides, there would be addition of another primer on the direction of which the DNA polymerase 3 would be adding nucleotides from 5 prime to 3 prime. So after the addition of primer by the primase, again another DNA polymerase 3 enzyme would be coming over here, and that would be adding the nucleotides from 5 prime to 3 prime till here, and so on. This will go in that manner. Now if we talk about this strand over here, there will be the formation of primer once again, there would be the formation of primer. Okay, there would be the formation of primer over here as well. Let's say this is the primer. Now over here the primer would not be added from this side because this side is 5 prime to 3 prime, and what we really need to create is an anti-parallel strand, and that anti-parallel strand would require the primer to be added on the opposite side from 3 prime to 5 prime, because primer is from 5 prime to 3 prime, and the DNA would be created in the direction of the primer, due to which the anti-parallel strand would be required to be created from the anti-parallel strand, and the primer would be required to be created from 5 prime to 3 prime direction, due to which we are going to add primer at this end rather than from 5 prime to 3 prime strand. So when primer would be added from over here, what actually happen is that, sorry, we have again made a mistake, that over here primer would be added from this part. Now primer would be added anti-parallel, such that this is 5 prime to 3 prime, primer would be added from 5 3 this is 3 prime to 5 prime, primer would be added from 5 prime to 3 prime. Now it is added on the opposite side, so that the strand which is complementary daughter strand which is formed is anti-parallel. So that anti-parallel strand which would be formed would be a would be, formed after by the DNA polymerase 3 by the addition of nucleotides, and once addition of nucleotides occur, after which there is another primer coming which is from 5 prime to 3 prime, after which another strand would be there, another primer would be there, and so on. This is how the elongation process would be continuing. Now over within this elongation process, within this elongation process the strand which is the complementary daughter strand which is being produced from 5 prime to 3 prime direction is continuous, because it is not having primer in its direction. It is having primer in its direction, but that is forming a continuous strand which goes in this direction from 5 prime to 3 prime, and that is not having the primer in its direction, due to which this strand is supposed to be a continuous strand. But if we talk about the direction of but if we talk about the direction of the complementary daughter strand which runs from 3 prime to 5 prime, that strand is having um, the strand is having a primer in its direction. That strand is having a primer in its direction, due to which and that strand is having a primer in its direction, due to which it causes to create a discontinuous strand, because this basically forms the 5 prime to 3 prime complementary daughter strand is having a continuous it is going continuous because there's no primer in the interval. But if you talk about 3 prime to 5 prime strand, it is discontinuous because it is having several primer coming at its interval, because it is in the anti-parallel direction from 3 prime to 5 prime, which is anti-parallel to the direction of the strand which is from 5 prime to 3 prime, anti-parallel to the direction of the parent strand which is from 5 prime to 3 prime, due to which the 3 prime to 5 prime strand over here, which is discontinuous, is known as the lagging strand, while the strand which is from 5 prime to 3 prime over here, the strand which is from 5 prime to 3 prime over here, this 5 prime to 3 prime over here, this strand is continuous, which is known as leading strand. Now the leading strand would be having its complementary parent strand running from 3 prime to 5 prime, due to which we can call this parent strand as leading strand template, which is from 3 prime to 5 prime. Likewise, if we talk about lagging strand, the lagging strand would be having its parent running from 5 prime to 3 prime, due to which we consider the lagging strand to be having its parent strand as lagging strand template, because it serves as a template for the lagging strand, due to which we call this as lagging strand template. And likewise, leading strand template serves as a template for the leading strand, due to which it is known as leading strand template. Now, one more thing that I like to highlight over here is that over here the direction which we are following is from this side. We are following direction from this side, due to which it is being considered, the leading strand is being considered from 5 prime to 3 prime, and the leading strand template is being from 3 prime to 5 prime. Likewise, the lagging strand is being considered from 3 prime to 5 prime, and the lagging strand template is being considered from 5 prime to 3 prime. So this is how elongation process occurs. Now at the termination phase, what actually happens in termination phase is that there are certain enzymes which are known as DNA polymerase 1 and 2. So DNA polymerase 3 has the capability to polymerize DNA, causes polymerization. DNA polymerase 3 has the characteristic of polymerization. Polymerization character is for DNA polymerase 3, DNA polymerase 2, DNA polymerase 1. All of these are having polymerization capability, but why do we utilize DNA polymerase 3 in polymerization process? This is because DNA polymerase 3 is having the fastest rate of addition of nucleotide, due to which it causes to speed up the process of replication. It adds 1000 nucleotides per minute. 1000 nucleotides per minute. On the other hand side, the DNA polymerase 1 causes to add 100 nucleotides per minute. 100 or 50 nucleotides per minute, due to which the least rate of addition of nucleotides is by DNA polymerase 1 and the highest rate is by DNA polymerase 3. Now when it comes to uh, DNA polymerase enzyme, it has got two properties. First is polymerization and second is exonuclease property. Exonuclease is an enzyme which causes to break the bond between the already formed, which causes to break the hydrogen bond between the sorry exonuclease, which causes to break the hydrogen bond which is present between the two strands of the newly formed DNA molecule. Now, in order to break the hydrogen bond of two newly formed DNA molecule, that property is known as exonuclease activity, such that in order to break the newly formed hydrogen bond of the newly formed DNA strand in order to proofread or in order to rectify any error in replication is done by a special enzyme which is known as exonuclease. All of the three DNA polymerase enzymes are exonuclease for strands from 3' to 5'. But when it comes to the property of exonuclease for but when it comes to the property of being an exonuclease for the strand 5' to 3', that is only possessed by DNA polymerase 2, due to which in proofreading of 3' to 5', all three DNA polymerase 1, 2, and 3 take part. But when it comes to the proofreading of the 5' to 3' strand, then there is only DNA polymerase 2 taking part. So this is the major difference. Now, what actually happens in termination phase is that what actually happens in the termination phase is that uh, the DNA polymerase 2 and DNA polymerase 1 act for the proofreading of the DNA, act for the DNA polymerase 1 and 2 act for the proofreading of the DNA and rectify any sort of error which is made during replication. And alongside that, uh, rectify any sort of error which is made during replication. Alongside that, the within the structure which we have studied, we have seen that between these structures, these DNA fragments, the strands which are formed are not the complete strands. Rather, these are DNA fragments of 100 to 200 nucleotides or 1000 to 2000 nucleotides. These fragments, which are of 100 to 200 or 1000 to 2000 nucleotide fragments, are basically known as the Okazaki fragments. These Okazaki fragments are now need to be joined together, combined together in order to form a continuous DNA strand, which would be anti-parallel to its complementary strand. So for that, what we actually do is we utilize uh, an enzyme which is known as DNA ligase. DNA ligase causes to join the Okazaki fragments to form a continuous anti-parallel DNA strand, a continuous anti-parallel daughter complementary strand, and also causes to and causes to and also causes to add nucleotides in the region where there was the formation of primer. By removing the primer and adding certain nucleotides, form it causes to causes to add nucleotides into it and form a continuous DNA strand. By removing the primers, by removing the DNA polymerase and forming and joining the Okazaki fragment, it causes to form a continuous DNA strand. So this is the process which is occurring in termination phase, and the formation of Okazaki fragment occurs in elongation phase. This is all about the process of replication. Hopefully you enjoyed the video. We'll meet again in another video.